All right, my girls. So hopefully, on Tuesday, you guys got a good introduction to transcription and translation, and how the DNA actually gives us the proteins that we've been talking about since the beginning of the year. So this is kind of, I guess I'd say it's a longer PowerPoint almost. It's not necessarily that it's that long, but there's a lot of information in it. That's just going to take a while for us to get through. So this will probably end up being quite a few screencasts. I'm not quite sure how many yet, because I don't really know how fast I'm going to be able to get through it. But I'll do my best. I'll try to stop at natural stopping points before I continue on to the following slides if I'm about to run out of time. So just bear with me. Like I said, I'm learning how to do all of this too. So let's get to it. So the first thing I want to talk about is the central dogma theory. And if you remember what we've been saying all year long about theories, is that they're oftentimes backed up by a ton of scientific evidence. They may just not necessarily be proven. So remember, we don't prove things in biology. We only support them. We only support our hypothesis. We only support our ideas with all of the evidence that we have to support. And the central dogma theory, one of the main theories, kind of like the cell theory, it's one of the main theories around all of life, around biology. And there's a lot of evidence to back it up, a lot of studies that show that this is the way the information eventually makes its way from the DNA into the protein. So it's really the center of what's controlling all living things in biology. And basically what the central dogma theory says is that the DNA is going to have the information to make proteins, but that DNA is going to have to first be transcribed into messenger RNA through a process called transcription. So that's this guy over here. It's the first thing that's going to happen, is the DNA is going to be transcribed into RNA. That messenger RNA can then leave the nucleus because it's no longer DNA, it's now RNA. And it can leave the nucleus and go to the ribosomes. And if you remember way back from the beginning of the year, the ribosomes are where proteins get made. Okay, so the ribosomes are going to be where the RNA is made into proteins. So that is, that is step 2 over here. Okay, so this is the central dogma theory, that DNA will be transcribed into RNA, which will then be translated into protein. Okay, central dogma theory. So I wanted to remind you guys a little bit about proteins and the structure of a protein and the monomers that make up proteins. It's been a really long time since we've talked about this stuff. Oh, okay, Nil and Buddha, just chill out. That was my dog and my cat. I'm so sorry. Anyway, arguing over nothing, should I add. Okay, so anyway, it's been a really long time. Really, it's been like since before Christmas, and really, it's been like since before or right after Matt's accident that we actually talked about proteins. So I just wanted to come back and give you a couple reminders in case you forgot. Remember that the monomer for proteins are amino acids, so the building blocks are amino acids. There are 20 amino acids. Of the 20, there's a certain number of them that are considered essential. I can't remember. I think it's 12 of them are considered essential. So you have your essential proteins and you have non-essential proteins, or I'm sorry, essential amino acids and non-essential amino acids. Technically you need all of them, but what we're trying to say when we say essential versus non-essential amino acids, the essential amino acids are the ones that you have to get from food. You can't just make these inside of your cells as you need them. Now, the non-essential amino acids, we have the building blocks in our cells to make those amino acids without having to eat anything to get them. But most of the amino acids are considered essential. We have to get them from our diet, which is why it's really essential that we eat things like meat. And if you don't eat meat, if you're vegetarian or vegan, you need to eat a lot of like beans, red beans, white beans. Any type of bean has a lot of protein in it. But because, again, we can't make all of those amino acids on our own. The order that those amino acids are placed in is going to determine what the protein is and therefore what the protein does. Because remember, structure equals function. So the order of the amino acids is something that we call the primary structure. Primary structure. And that, again, is just the order of the amino acids. And you should be familiar now with the different amino acids because you used the codon chart on Tuesday in your assignment. So you should be familiar with methionine, that's the start codon. Some of the other ones, you saw like the SER, that's serine. You saw the THR, that's threonine, the ARG, that's arginine. There's just a bunch of them. They all have these crazy names. But those are the amino acids, and the order of those is going to determine what the protein is. Now, don't forget our functional groups that make something an amino acid. Remember, we have the amine group, so that's the NH3. So if we look at our picture over here, that's this guy. Or if we look right here, it's this guy. Okay, that's our amine group. And then the carboxylic acid, or the COOH group, the COO group, if you remember that. And that's the part that makes it an acid. So amino from the amine group, and then acid from the carboxylic acid. So here is the COO. And the H is gone because it's showing that it has already bonded with another amino acid in line. That's just not being shown, but that's why it has this minus instead of a hydrogen. But it was here before. And then, okay, can you please move, Khaleesi? My students do not want to hear you hiss and meow at the dogs. And then here on this guy, here's the COOH, or the carboxylate group. Remember, the carbon is right here in the middle. These two lines represent that it's double bonded to oxygen. And then the OH group over here, that hydroxyl group. All together, that is the carboxylic acid group. And then remember, the thing that makes each amino acid different from the others is the R group. The R group is going to be what makes the amino acids different from each other. They're just different chemical groups. They can be rings, they can just be branches. They can really take on lots of different shapes, but they're just going to be a different, the different part of the amino acids. It's what they do not have in common. Remember back to when we did biochem, there were some that were acidic, there were some R groups that were basic, there were some R groups that were polar, there were some that were non-polar. Remember, if something's polar, it likes water. If it's non-polar, it doesn't like water. So there's just a bunch of different R groups. And then the last thing that I want you to remember from biochemistry when we talked about proteins is that the amino acids are put together by dehydration synthesis. So that's the removal of a water molecule to put things together. So in this case, we're making a peptide bond, which is just a fancy name for the covalent bond that is formed between two amino acids. Okay, and essentially the water is going to come from the amine group, so one of the H's from the amine group, or two of the H's from the amine group, actually it'll just be one, and then the OH from the carboxylic acid group. Okay, that OH will be removed. So an OH plus an O, that's H2O. That's dehydration synthesis. Okay, so I think I'm going to stop this video here, and I'll start my next one with the players in protein synthesis.